especially in the context of omics technologies and deep phenotyping. It's really the cost of these measurements that has gone down. Just this past summer, the cost of sequencing a human genome, the whole genome, dropped by 40%. It dropped from $1,000 to $600. We expect these prices to continue to drop to the point where they're getting closer to the point where they can be included as part of routine clinical care, as opposed to only for research studies or clinical trials, that sort of thing. One of the most exciting developments, particularly relevant to cancer, is the development of new single-cell technologies. Now, single-cell technologies themselves, and I'm talking about, instead of profiling populations of cells, I'm talking about measuring an individual single cell and being able to measure different compounds and molecules within that single cell. Those kinds of technologies are not new. Rather, it's the omics dimension. It's the ability to profile at the genome scale and to be able to measure thousands of DNA transcripts or dozens of proteins or on the order of a dozen metabolites. Those are the technologies that are really improving. And with respect to cancer, these kinds of technologies are particularly important because of the heterogeneity, and particularly the intratumoral heterogeneity. So again, this is this idea of within a single tumor, there's different subpopulations of cells, and these different cells have different properties. They might have different metabolic functionality, they might have different resistance to different types of drugs. And so if we're able to use these developing single-cell technologies to better understand how each of these single cells are functional, what their different phenotypic properties are, that just allows us to develop even better therapies and to really start to understand then how can we personalize the therapies down to each person's cancer. If we can take a sample from a tumor, learn as much as we can. If one drug only kills 70% of the cells in a tumor because there's multiple subpopulations, single-cell technologies are going to help us get past that to 80, 90, 100% of effectiveness of different therapies.